Ok, so a gente pode recomeçar. Any of you have any question from yesterday that or topic to discuss? Anything? No. So today what I decided to do is kind of, so yesterday I gave sort of conceptual framework on aspects of resistance, right? Someone was asking about what is resistance, you maybe? You, someone. Versus tolerance, classical tolerance, and then also disease tolerance that is related to the plant biologist form of disease tolerance. Are these concepts clear, or should I rephrase some of them? I think resistance is very clear, right? Resistance is the control of pathogen load, basically. Tolerance, classical tolerance is regulation of immune response to self or non-self. So regulation of immune response to self is called central tolerance. That's because it's induced early in development in the thymus. Regulation of immune response to non-self, it's called peripheral tolerance because it's induced in the periphery when you contact microbiota, diet, and so on. Disease tolerance is the tolerance to the own immune response that is induced in response to an insult or a pathogen, or the tolerance to the pathogen themselves. So these are the three conceptual frameworks. So what I'm going to do today is to show examples of how we approach that in the lab. It's a little bit how we approach these three examples in the lab. So I'm going to give an example of each one of the three. Resistance, tolerance, peripheral tolerance, and disease tolerance. Is that okay? I try to make it very broad, but if I get too much into immunology, jargons, CD 155, you guys can stop and say, you know, I don't understand what is a CD. Okay? So it's okay to say, I have no idea what you're talking about. Then I try to make it less specific. Is it light? From okay, because there are some images here, so. All right. So part of the lab is studying the concept of resistance at the epithelial layer of the intestine. So as I was saying yesterday, the epithelium of the intestine is a single layer of epithelial cells that is the largest area that we are in contact with outside environment, even though the gut is inside. The skin, as I was saying yesterday, is 100 times smaller than the gut because the intestine has the villi, these structures that are present in the small intestine particularly, that go from the crypt to the tip of the villus. So crypt is these regions that invaginations, and the villus is these regions. And you have these microvilli, this brush border is called, these structures that increase the area of absorption for nutrients. So in the perspective of the immune system, is the largest area that we are in contact with non-self, right? So that's very important to have both resistance to the occasional pathogens that are trying to invade and create problems, salmonella and so on, but also an area that we should develop tolerance, because if we develop an exaggerated immune response in this layer, we lose this layer of epithelial cell, and that affects our nutrient absorption and so on. We lose epithelial cells, it's very serious. Surprisingly, the main T cell population in the body, you guys know what is a T cell, right? Most of you. Anyone doesn't know what is a T cell? So the main site of T cell, the main population of T cells are cells that live in the epithelium of the gut. These are called intraepithelial lymphocytes. So this is the most number, in terms of numbers, the highest number of a T cell subset we have. And that's because there are about one of these intraepithelial lymphocytes for every 5 or 10 epithelial cells. Epithelial cells, again, if you have a 300 square meter, a tennis court covered by epithelial cells, and you have one T cell for every 5 or 10, That makes the number of hundreds of millions of cells that you have in mice. In humans is also the case. So the lab has focused on understanding what these cells are doing in the same perspective that I was saying earlier. How can you control invasion of a pathogen while not developing exaggerated response to microbiota and diet? So that's a balance between tolerance and resistance that I was mentioning yesterday. And these are some of the studies we did kind of describing what these cells are doing and so on. But I want to focus in one study particularly from Bernardo Reis. He's also trained in biology at UFMG. And he has been in the lab since we started at Rockefeller 12 years ago. And these are several people that have worked with Bernardo in some of these studies. So this study was actually published several years ago, but I like to give this example so you kind of see types of things that we can, the way we can ask a question in the gut epithelium. So these are the first 2 images done, live images in mice in the intestine. So Bernardo established this in the lab. We're using multi-photon or 2-photon microscopy. So that allows the microscope to go deeper in the tissue. So the refraction of the laser can go deeper because there's 2 lasers pointing, so you can correct that, go deeper in it. And then you can image intestine of a live animal. You can also eventually do that in humans doing endoscopy, for example, or colonoscopy. But that's in mice. And of course, the mice are anesthetized for that. This left is an image of a gut of an animal at steady state. So we don't do anything. Just take the animal and image the intestine. And in green, you're going to see this intraepithelial lymphocytes, a major subset of this. Doesn't matter. It's gamma delta T cells. Doesn't matter what type of T cell. It's a subtype of T cell that is particularly enriched or present in the gut epithelium. So the first surprise when we did this image is that these cells are moving. So so you see this, each one of these green cells is a T cell, and each one of these structures is a villus of the intestine. We have many villus, villi, and then the cells are moving. And they are moving in a way that's pretty interesting. I don't know why I stopped. Let's see. They are moving in a way that's very interesting. They are going around, there's a basement membrane that separates the core of the body from the epithelium. And they are moving above the basement membrane, along and underneath the epithelial cells, which are in purple. I think the light here is too strong, so you can't see very well. And they are moving basically in this way. They are moving downwards in spiral. The villus is like this. So let me go back to the villus. The ILs, they are moving like this. Of course, this is a scheme, right? The epithelium is completely covered by the epithelial cells. And they are going around elliptically downwards. The first surprise that they were moving, so we didn't know that they were moving. The second surprise, and it's very interesting for physiology, is that they are moving downwards like this at the speed that is exactly the opposite of the speed that the epithelial cells grow upwards. So here at the base of the crypt of the villus, you have stem cells that differentiate from stem cells to the epithelial cell subsets. Right? So you have stem cells here. They divide. This region here is called transit amplifying region. So the cells divide as they differentiate from epithelial cells. And then they grow upwards. So you ask, oh, what happens when they reach to the tip? They fall. They fall into the lumen. So the entire epithelium of the gut in humans and in mammals is completely changed within 5 days. So today you have these cells in your gut. 5 days, 1 week from now, it's completely different cells you have there. That's because the villus grow from bottom to top. And the ILs are kind of moving physiologically downwards so they don't fall into the lumen. So it's very interesting, this dynamic. But that's just doesn't matter. What happens when you infect an animal with a pathogen? So here we gave Salmonella typhimurium, which gives the salmonellosis in humans. And then we looked how this movement changed. That's the only question we had. What changed this? And what you see is this change. It's very clear. You can see. Now, they are no longer moving around the epithelium, like in straight movements. They are now going a lot between epithelial cells. Do you see the change, or should I show again? You see, right? They are not going this anymore. They are visiting two epithelial cells. So it actually is the only word we created in science so far. It's we called flossing, because like flossing, the free dental, right? You do this. And that's what they are doing. They are flossing the two epithelial cells. And that is a kind of striking change in movement that these cells have. Okay. And I'm not going to show every detail of this imaging. But the interesting comes from the quantification. So what we estimated of the steady state movement of the cells is that they are moving around the villus, right, like this. And if you do in 30 minutes of image, you kind of accumulate the tracking of these cells, you see that they make the contour of the villus because they are covering the entire villus. So what we estimated is that these cells can cover these gamma delta T cells, the entire epithelium of the intestine within 2 hours. That means every single epithelial cell is touched by this T cell every 2 hours. But there are two types of ILs, gamma delta alpha beta. So it means, and there are double of cells that we are imaging here. So that means that every epithelial cell encounters a T cell every hour that we are living. Yes. Okay. Oh yeah, the microphone.
I was just going to ask, what is a villus?
What is a villus?
Yes.
So villus are these structures that are the shape of the intestine. So I think I have even an image of that. It's maybe difficult to find. Let's see. It may take a few seconds. Should I try to find? Because I think that will explain very well. It's just, I don't know if it's going to be easy to find. Because that shows exactly what is a villus. Keynote. Let's see if I find that video. If I don't find, I stop. I found. So here you have the gut, the tube, right? The intestine. You cut open the gut. If you do that, I mean, didactically, you open, and this is what you see. Each one of these structures is one villus. That's what the intestine is made of. Millions of villus, or villi if it's plural, right? Do you see what I'm talking about? See, this is one villus, and you see more. You see the epithelial cells. Each one of these is one epithelial cell, you see? And these are the kind of the circulation, the vessels. This is the stem cells I was talking about, and they grow in this direction, transit amplifying compartment, and they go upwards to the tip, and then when they reach the tip, they fall. You exchange, they die. It was clear? Okay, that's what makes the intestine so absorptive, because you increase the area by doing invaginations. Okay, it's not every answer I have, every question I have answered that is so clear.
Yeah, I have a question.
Yeah.
Where do the T cells come from?
You can say I repeat. Okay.
Okay. Where do the T cells come from? From the lumen, from the bottom, and then go up?
So that's very interesting. So, in fact, if you guys want to make more dynamic, you can ask and I repeat, because I can repeat very quickly. I think it's easier. Okay, so where the cells come from, not developmentally, but locally. So, and it's not known. But basically, I would have to come back there. Oh, let me go here. It's not clear. One thing is clear, and I may show some of that in a few slides, but once the cell gets activated, I was showing yesterday, remember the retinoic acid example, that dendritic cells secrete retinoic acid, make the T cells migrate to the gut? The lab has asked this question that you ask, and we have studied this study here, Angelina, no, this one, Maria London, in 2021, that we kind of addressed a little bit of that. They get activated in the gut draining lymph node, which is the topic of the next example. They get activated. They get directed via the dendritic cells to migrate to the intestine via retinoic acid. They migrate to the intestine. When they reach the intestine, they can't go, we think, like structurally, they can't go to the epithelium direct. So they go normally to these regions called lamina propria. And what we found is that some cells stay very little. They just stay here and then migrate to the epithelium. Some cells stay longer. So for sure they have to fall here because that's where the lymphatics will end up, and they will extravasate to the tissue. And then they may fall maybe closer to the epithelium, or they have a program that is close, or they spend more time, then go to the epithelium. So that's kind of, you can ask, I repeat.
Okay, and after the movement of going down, they just die?
So that's the issue. That's what I was saying about the way they grow. So the question is, what happens once they are moving? So there are different types of T cells living there, and they have different lifespans. Some cells may live for years. But the reason that they can't do that, because they are going downwards as the epithelium grows upwards. So they stay in the same place, basically, right? Because the epithelium is growing, and they are going down. So it's it's very interesting for this reason. Okay, all right. So we estimated all of this and calculated how they move, we calculated the distance that they go from this region here to the flossing behavior. And we associated this with a variety of functions. So what we found, and I'm not going to show all the data here, is that this movement, when they change the movement and start going between epithelial cells, is very important for the protection against the salmonella that is invading between epithelial cells. So this change in movement, it's important for the protection against the salmonella invasion in this model. I'm going to show two examples of things that we did in calculating that. So here, on the left side, it's a salmonella that is red. It's not a single salmonella, it's a group of salmonella that they grow, proliferate, in red here. And in green, it's one of these T cells. And you can see some of the cells they have a bias in migrating to regions that the salmonella is invading the epithelium. So some of the cells are inside an epithelial cell, and we can see the change in dynamics of the T cell, and we can calculate over many cells that they are growing closer to the salmonella. It's very interesting because salmonella, going back here, salmonella accumulates at the crypt region. So when there is infection, the T cells, your question, they go faster downwards. So they reach the crypt in a much faster pace than they normally would. And then this movement downwards and the change in movement that they have for the flossing, it's important for the control of the salmonella burden. So that's a mechanism of resistance. On the right side is the toxoplasma cyst. So it's much larger, but we also estimated that for toxoplasma. That's not intracellular, that's extracellular, but we calculated this movement around these infected regions. And it's part of the protection mechanism by these T cells. But what is interesting here, and I want to, again, I'm not showing every single data, but you can ask questions, is that how do T cells know that you have an infection? So there are two possibilities. That's why I'm showing this. Possibility one is that the T cells can sense the pathogen. That's possible. They can sense the pathogen. Possibility two is that the environment tells the T cell that there is an infection. And we looked for the two possibilities in this study. So what we did, we knock out the sensing mechanism of pathogens. They have a hub, which is downstream of toll-like receptors, doesn't matter, called MIghty88. It's a hub that synergizes a lot of the sensing pathogens that cells have. And then they go here. we knock out from the epithelial cells and we knock out from the T cells. What we found is that the epithelial cells sensing of the... So I have a question. You guys that are in the computer, you are looking for something else? Just for me to know. Just because I know that we traveled a lot distance to give a class, and if you guys are looking things that are not related to the class, you can leave. I have no problem in leaving. But I do want you guys to pay attention. And I have no problem. If you're looking for if you're looking for a paper that we published, that's okay. But if it's unrelated, you can leave. And I wait for you guys to leave. I have no problem at all. Zero, it's okay. It's just because, the carbon footprint of me coming here is a lot for you guys to be, social media or whatever you want. I have no problem with anyone leaving. Seriously. I'm not going to say anything. It's okay. Okay, so if you are here, pay attention or leave. Because, it's a lot of travel to come here. So what we found is if you knock out the gene that is sensing pathogens in the epithelial cell, you abolish the change in T cell response to the pathogen. So that means that the T cells require the communication with the neighboring epithelial cells to respond to the pathogen. And there is more of that. For those that are interested in metabolism, we found that the increase in glycolysis of the T cells is essential for the cell's change in behavior. So what we found is that how the T cells change the metabolic profile, the way they utilize energy, is via communication with the epithelial cells as well. So if the epithelial cells cannot sense the pathogens, the T cells do not change their metabolism and their movement behavior. So what we found basically is a communication between epithelial cells and T cells that can tell the T cells that there is an infection, and they change the way they utilize energy, and they also change the way they respond to the pathogen. And this change in behavior is essential for preserving the epithelial cells' health status during infection. So if the epithelial cells cannot sense the pathogen, the T cells cannot sense that there's a pathogen there, and then there is invasion of the pathogen. So there's a communication between the epithelial cells and the T cells, and that's what we described here. And any question on this? Yeah. You can say I repeat, so it's fast. Anyone can ask without the microphone, and I just repeat.
I think it's okay. Yeah, it's
okay.
These T cells, they have specific functions. For instance, they are helpers. They are cytotoxic.
Yes. So I didn't get into this detail because I didn't want to get too much into immunology. But you're asking, what is the function of the T cells? These ILs are both cytotoxic, highly cytotoxic, and they also express a bunch of molecules related to response to intracellular pathogens like interferon gamma and so on. So they are, I would say, what's called TH1-like and cytotoxic. However, you may ask, well, if the cells are cytotoxic, means they kill the neighboring cells, why they are not killing the epithelial cells at steady state? So it's very interesting because these cells have what's called NK inhibitory receptors. NK cells, which are involved in, for example, in transplant, rejection, and so on, NK cells sense the absence of MHC class 1, and then they kill the cell because they have a mismatched MHC. These cells have the same inhibitory receptors NK cells have, and so they are not constantly killing the neighboring cell unless these NK inhibitory receptors are downregulated, and then they are able to kill an infected cell. And this is mediated via cytokines IL-15 and so on. So they are cytotoxic, but they're not killing at steady state. And this switch between cytotoxic and cytokine producer, it's very important in diseases like celiac disease and so on. So there's a very interesting pathway there. Another question that is related to what you asked, and I'm not going to show the data unless someone is interested, I can show later or so on, is if you notice what I just presented, is the communication between epithelial cells and a T cell. What happens in tumorigenesis in cancer is exactly the same. So you have a cancer in the gut. It's a transformation of the epithelial cells that becomes hyperproliferative and create a polyp, then creates a tumor, then extravasate metastasis, and so on. So what we did recently is to understand whether these same cells that are intraepithelial lymphocytes are playing a role against cancer. And that's also work from, this was just published a few months ago. And I'm not going to show this publication, but basically what we found is that these intraepithelial lymphocytes that are living there at steady state, that are cytotoxic, your question, they are essential, very important, I don't know essential, but they are very important in controlling early tumorigenesis. So they are able to detect and kill transformed cells at steady state. So if you target the cells, if you delete, you have more tumors in different models. However, the tumor, as the tumor grows, it changes the microenvironment. And then also change the type of T cells that are there. So you accumulate some T cells, another subset of intraepithelial lymphocytes, that are helping the tumor to grow. So there are other subsets of IELs that are not cytotoxic, that are producing cytokines that are pro-tumorigenic, so they help the tumor to grow in size. So that's the study that we looked, that's kind of analogous question to what we addressed. Yeah? Yeah, you can ask, I repeat.
Yeah, I have a question regarding the response of the T cells to the salmonella in the cryptosome and the interaction between the epithelial cells and the T cells. So just to contextualize, I remember that in the inflammatory response in a vessel, the lymphocytes interact with the endothelial cells through caderins,
That's right.
intercellular molecular adhesion.
Yeah.
So I was wondering if these T cells, the movement, it's related to the expression interaction among with these molecules?
Yes, that's right.
And if there are any proteomic and genomic studies that explore that?
Yeah, no, that's a very interesting question. He's asking how is the cell-cell contact mediating this change in movement or residence of the cells? And indeed, it's exactly what you said. So it's a long explanation, but very briefly, the cells to go to the gut, they express gut homing receptors. And the question that I forgot now who asked. You asked about where they go. So once they arrive in the lamina propria, they express a group of integrins, alpha-4 beta-7, that binds MEDCAN in the stroma cells of the lamina propria. And then to go to the epithelium, they downregulate alpha-4, upregulate another integrin called alpha-E that binds ecadherin, which is a cadherin, another form of integrin, expressed by the epithelial cells. And this change in dynamics of cells also involved additional forms of adhesion molecules like claudins, occludins. So it's all mediated by that. In fact, someone asked exactly this question and how this deficiency in occludin would affect this behavior and indeed affect. So it's exactly via this dynamic expression of these molecules that the cells move there and they change movement.
Yeah, just to add, I studied toxoplasma gondii infection, and I saw that pattern that some cadherins and integrin molecules are involved in the interaction with the pathogen, the invasion, and also the response of the T cells and other inflammatory cells against the pathogens.
Exactly. So toxoplasma is very interesting because there is a component of the CNS invasion that they have to cross blood-brain barrier to go there. There's a component that is intestinal. That's kind of what I was showing on the cysts, right? And this all involves cell-cell contact for the invasion of the cyst, depending on the stage of the parasite. Yeah, it's very interesting. Okay, so I'm not showing this study here, but if you guys are interested, you can look. But that's exactly showing this dynamic of intraepithelial lymphocytes. So another form of resistance is resistance against transformation in cancer. And that's kind of, you can imagine that the same way that they react against an invading pathogen, they should react against cancer to form. That's a classical form of surveillance. I don't know, surveillance is a little bit controversial, but this is an example of a T cell detecting some change in the state of the tissue and kind of resolving. All right. Any question for this part? I'm going to give the second example now, which is tolerance.
Why is surveillance controversial?
It's controversial because surveillance was proposed in studies using RAG knockout mice that are deficient in adaptive immunity. And these studies have been redone in animals with full immune system. And it's much more complicated or complex than just Immune system is there, is defending against cancer. I was just giving an example of why. When I was asking the function of these intraepithelial lymphocytes, there are intraepithelial lymphocytes that indeed perhaps can detect transformation, and some that actually are helping the tumor growth. So immune surveillance has to be, it sounds like the immune system is surveilling in detecting tumors, and it's not clear because in many cases it's helping the tumor. So this is part of the explanation why it's controversial. A form of surveillance exists, but we are observing a phenomenon, right, and saying this is happening in our interpretation of the system, right, and kind of teleology, sort of assumption of attributing function to something that our experiment can only give an answer, limited to our question. It's a philosophical discussion. Yes. Yeah.
Is this related with some type of differential dynamics of these cells across the gut?
So the question is, the toll-like receptors are the sensors of pathogens. And you are asking whether, for example, the TLR, the sensors, they have interesting dynamics. For example, some TLRs are not expressed in the apical. This is the apical side. They are only expressed in the basolateral. Why? Because in the apical, you have constant interaction with pathogens. So if you have sensing all the time, you may trigger inflammation. So some are expressed in the basolateral. So when you have invasion, disruption, you can activate sensing mechanisms, right? And you're asking whether there's a dynamic change, expression along the GI tract or within the epithelium, and whether this is related to this movement. And we don't know this. In fact, it's something that we would like to test this, but it's a little bit tricky to modulate toll-like receptor expression in only a segment. But it's an interesting possibility. Okay, so yes.
Is that response the same for virus and for bacteria?
Very different. But what we are. So the question is the response similar between different pathogens? And it's very different. I was mentioning yesterday that parasites, helminths, for example, trigger a type 2 response that is similar to allergy. Viruses, similar to intracellular bacteria, trigger sort of a type 1 immunity. Extracellular bacteria trigger a different type of immune response. But sometimes these responses are not well directed, and you have kind of pathological inflammation. So that's a summary. But we have tested different enteric viruses that also trigger this movement. But the mechanism could be different in terms of communication between epithelial cells and T cells. So I'm going to give an example of peripheral tolerance. And that should trigger a lot of discussion, I guess, because that's a very interesting concept. Yesterday, I presented this slide that is exactly showing the different types of T cells, right, that we are just discussing here. These are forms of CD4 T cells, but you have parallel to that, to the CD8 T cells, also gamma delta T cells. But these are sort of states of differentiation of cells that they can perform different functions. So the T helper cells, CD4 cells, what they do is to help other cells to modulate. They help macrophages, they help dendritic cells, they help B cells. Without T cells, B cells cannot secrete better antibodies, for example. In any case, a concept that was very kind of prohibited in the 80s, 1980s, was the existence of cells that are not inflammatory but actually are regulatory. They are suppressing immune responses. So the existence of regulatory T cells. And these are cells that now we know that mediate part of this peripheral tolerance that I've been discussing, the way that you control inflammation against antigens that are not self, but they are not dangerous. They are harmless types of interactions. In fact, you have to control even immune response that are against pathogens as well, part of this third topic. So here is a slide explaining one of the many mechanisms that explain how can you react to a commensal or to diet differently than you react to a pathogen. So how is the T cell knowing. The T cells normally don't sense. You ask about toll-like receptors in the right way. Toll-like receptors normally are expressed, the sensing of the pathogens is expressed by what some people call, like Ruslan, accessory cells. Dendritic cells, macrophages, epithelial cells. These cells sense the pathogen and can communicate that there is a pathogen or not to the T cells. So this slide is very simplified and is only showing one example of how this distinction can be done. Commensal bacteria versus pathogenic bacteria. Of course, you can transpose that to viruses, enteric viruses that are endogenous. There are several that are not causing disease, and enteric viruses that are pathogenic. The same thing is true with helminths and so on. So I think Carolina mentioned yesterday very briefly how a T cell communicates with an antigen-presenting cell. And you guys learned that in basic immunology as well. But this example here is kind of what people think that explains in the intestine with so many commensal bacteria, why you're not reacting and causing inflammation with the bacteria. It's because the bacteria cells, in the context that the commensal bacteria have, they don't induce upregulation of these co-stimulatory molecules in the dendritic cells. Dendritic cells are the ones that activate the T cells. They tell the T cell that this is a context of inflammatory. If the dendritic cell is highly inflammatory, it will induce the inflammatory T cell. But in the steady state, normal homeostasis of the intestine, this is not happening. So the level of co-stimulatory molecules is very low. You guys may be familiar, maybe you don't know co-stimulatory molecules, but you know cancer immunotherapy. So cancer immunotherapy target exactly this communication that I'm talking about here. Because the T cell, for example, if the T cell is communicating with the dendritic cell that is expressing PD-1 or PD-L1 in the T cell side, PD-1, PD-L1, this can tell the T cell that this to suppress the activity. So that's a strategy that tumors have to suppress immune response anti-tumor. The same is true with other co-stimulatory molecules like the cousin of CD28, CTLA-4. So the main immunotherapy is CTLA-4, PD-L1. So you're blocking the communication between accessory cell with the T cell the same way that the steady state intestine is happening. So as a result, the type of communication here is not inflammatory. You produce cytokines that are anti-inflammatory, IL-10, and we induce a type of T cell that's not pro-inflammatory. And that's the steady state daily routine of the intestine. So it's inducing a less inflammatory state of T cell. As opposed to when you have invasion, salmonella invasion, is sensed by and activates dendritic cell. These dendritic cells upregulate co-stimulatory molecules, secrete cytokines, like IL-12, then activate the T cell, and this activated T cell will then migrate to the intestine, to the site of the salmonella, and perform some resistance mechanism. Is that clear, this example? Yeah.
How would these distinction, let's say, be learned as you, let's say you're a baby and your intestine is being colonized?
Yeah, so the question, you can make it broader. How the dendritic cell knows, right? Yeah. And this is, the answer is very complex because there are multiple ways of knowing. For example, João, right? Yeah. João was asking that the toll-like receptors dynamic in the apical versus basolateral, it's regulated. So if you have toll-like receptor 4 that detects LPS only in the basolateral, normally a bacteria that is here is not activating. But if you have a bacteria like this that is coming to between the cells, activates because that's where the receptor is. That's one way. There are multiple ways of the detection. So if a virus is intracellular. So you have cytoplasmic toll-like receptors that if a double-strand RNA is present or CpG present in intracellular bacteria is in the cytoplasm of the cell, activates toll-like receptors. These are not in the extracellular, another way. And so on. Why, when we have a vaccination, coronavac, Well, CoronaVac doesn't work very well, but let's imagine that it worked very well. CoronaVac, you have an activated virus. Normally, you put an adjuvant, right? In vaccines in general, what they call the dirty secret of immunology is adjuvant. Alum hydroxide, CpG. CpG, what's the CpG? CpG mimics a bacteria infection and then makes the dendritic cell sense as a bacteria activates the immune system. So the adjuvant is a way to teach the immune system that is a pathogen interaction, pathogenic, so it's a context that will do that. So these are some of these examples of how this is done. In the development, it's the same thing. So as you are young, sometimes you have the expression of toll-like receptors is very different than when you age and so on. There's a lot of regulations in development as well.
So if I understand it correctly, very generally, it's not necessarily that you learn what is pathogenic and what is not pathogenic, is that our bodies have kind of mechanisms of, they understand what the mechanism of pathogenesis is, right? So if it's trying to invade, if it's, you have, you signal that and then you can say, okay, this is probably pathogenic.
Because the learn is teleology in a way, right? We are assigning something that doesn't belong to a system, right? I mean, we are thinking and saying the system learns, has a memory. So that's a complicated word because it's charged, right?
Yeah, I would
and I'm not criticizing.
Like a
we have to
network
use
that has
that's
reinforcement and things like that.
Yeah, but you can say that
clear here.
That's right, but you can say that the body expressing toll-like receptors only here. The body learned that shouldn't express in the apical, right? So it's a kind of anyway, it's a philosophical discussion. But yeah, I would say, but there is also the opposite. When you have an adjuvant, and that's important for the next discussion here, actually, is a good link. If the first interaction, immunologists like to use ovalbumin, the protein of the egg white, ovalbumin. You put ovalbumin with alum. Alum is the most common adjuvant used in vaccines. You put and immunize an animal. This animal normally will develop an inflammatory T cell that will turn into, in general, Th2, type 2 inflammation, and antibodies, and activated B cells, plasma cells. It will activate the dendritic cells. We will induce an inflammatory immune response. Next time you see ovalbumin, you can see without adjuvant because you already developed inflammatory memory cells that are steady state against ovalbumin. You don't need adjuvant anymore. So in that way you learned, right? It's a form of learning, part of immunological memory. That is not always true. And the example is next. Exactly next. Go ahead. Yeah.
My question follows the same path because, like, based on the previous model, we see that just the recognition of the commensal or pathogenic bacteria is through a molecular pattern, right? But then I want to know, like, how this mechanism works when you need to tune the population size of the bacteria, because it could have a commensal bacteria that goes an incredibly fast growth and then it becomes a dysbiosis. So how but the molecular pattern will remain the same. So how can we tune it to?
So the question of bacterial population size is very interesting and is not very well understood. There's a person called Bonnie Bassler at Princeton that studies that, but it's called quorum sensing of bacteria. It's a very interesting topic. But how quorum sensing, how a bacteria knows the size of its colony and how this is sensed is not really well known in mammalian systems. Your question, I don't think is understood. Your question is how can, whether expansion of a particular type of bacteria will be sensed. Of course, there is what is known. Let's say you may have E. coli expressed. We all probably have E. coli, Escherichia coli, right? And if E. coli is at a certain level, it's probably retained at the mucous layer and so on. If you have a bloom of E. coli, maybe the expansion of E. coli changes the presence of additional bacteria species that are controlling attachment to the epithelium. And then in the absence of this second bacteria, E. coli may be attached to the epithelium. And if E. coli attached, you have this enteropathogenic E. coli that maybe trigger the toll-like receptor. So this dynamic is better understood. How one species can regulate the other species and how this is sensed by the system. So this experiment is an interesting experiment. This experiment was done in the 70s. The phenomenon was known for about 100 years. So in the late 1900s, and that will depend on who is telling the story, but Native Americans were known to, do you guys know poison ivy? So poison ivy creates this rash in the skin. It's very bad, it gets worse. And one of the strategies, I'm not suggesting anything, is that people would ingest poison ivy and then they would be protected against skin reaction of poison ivy. That's one of the early description of oral tolerance. Nelson Vaz, who is Brazilian but was in the US, in fact, he was my undergrad advisor. Nelson Vaz, who was in the US in the 70s, did this experiment which became very important for the field later on. It's an oral tolerance experiment. So if you gave an animal, the example I just gave with adjuvant, but if you invert their order, okay? If animals eat a protein orally, just drinking or eat the protein, and you wait a few days, a week, and then you come with the adjuvant plus the protein, the same protein, there is mild to no response to this protein, as opposed to if you didn't eat the protein prior to the immunization, you have a strong response. So this difference here is called oral tolerance. There is a lot of interesting discussions and a lot is not known about this. But you guys understand the concept. If you do the opposite, if you immunize an animal with an adjuvant, like a vaccination, then you come with an adjuvant orally or nasally, you develop allergy. If you do orally, you develop food allergy. If you do nasally, you develop asthma. If you do skin inflammation. But if you do the opposite, if you ingest, you are protected against this inflammatory. So they think that this explains, even though it's an experimental model, why we are, most of us at least, not developing food allergies or celiac disease against gluten, because there is a daily routine process of tolerance that prevents when you have, like yesterday we were discussing why a particular antigen is allergenic. Remember? Someone was asking why, how do you know that this become, and I said, well, because of the lipid content, for example, of nuts. The immune system sees it as a however, if this is seen in a context that is not allergenic, you actually protect it. The example I also gave yesterday on this study on allergic kids that eat peanut butter before the development of allergy. So part of the lab is studying this phenomenon. In fact, it's a whole field, but we are very interested in this phenomenon because there are many interesting aspects of this. I'm going to show the experiment they did in the 70s. Yeah.
I have a question about the, I don't think I understood exactly what is the meaning of an adjuvant. It's just something that gives you an inflammatory response? Is that
I'm going to show one example.
Like
the readout, right?
Sorry?
You're asking about the readout. What are you measuring?
Yes, I'm asking about the term adjuvant. I haven't...
The term adjuvant.
Yes,
the term adjuvant, it's a molecule or a substance that you put in combination with the protein of interest to trigger an immune response.
Okay. So there are clinical trials of this in humans?
Oral tolerance?
Yes.
Or because adjuvant...
For allergies, for example. I have a milk or allergy. And if I don't want to have it anymore, I just make it with adjuvant.
So I'm going to give a Mineiro answer.
Okay.
So there are clinical trials, but there is a problem on clinical trials, although some work. The problem is if you immunize an animal, then you come with the oral antigen. You are inducing allergy. You're not protecting because that's a booster. You guys are taking booster of the vaccine. That's a booster without adjuvant. Oral tolerance generally works best when it's given prior to the inflammatory development of the. So what you're asking is, once I'm allergic, if I eat protein, well, normally it's allergy. You increase allergy.
Okay.
But there is a way to do that, that people, you probably know about, called desensitization. So that's, we think, is mediated via oral tolerance. So you give increasing small doses of the allergic substance orally, and then you kind of reestablish tolerance. So, you know, I'm giving people tried this with autoimmune disease in the 80s and didn't work. For example, you have multiple sclerosis is self-reactive T cells against the myelin protein, right? The myelin sheet. So if you give patients MBP, one of the proteins, myelin basic protein, orally, would you prevent or cure autoimmune disease? And didn't work. And one of the thoughts is that the inflammatory response was so developed that this is just a booster. Yeah?
Bel, are you going to say something more? No.
No.
In the case of milk, we drink milk when we are babies.
Yeah.
So is this, because I don't know, when someone is allergic to milk, I am like, oh, but you drink milk when you are a baby and you don't have an allergic reaction. So I don't know if maybe it's something about like, it's a milk from another species, but maybe, I don't know, thinking of oral tolerance and you receive some little doses of milk every day for a period of time.
Yeah.
So maybe you are, don't know, improving your tolerance of having an allergy in the future?
There are two components. One is there's a distinction between intolerance and allergy. So intolerance is when you can't handle a particular substance of the protein. For example, lactose intolerance, you're not developing antibodies to lactose. You just can't handle lactose. But there are indeed people that are allergic to milk. And your question of breastfeeding is very interesting because when you're breastfeeding, the milk has a lot of the components I was talking about, actually. The milk has high levels of IgA, which is unclear if it's important in mediating tolerance or not, but milk has huge amount of IgA, which are definitely important in the control of early infections and the way that the gut is colonized in the newborn. The milk also has many molecules, for example, TGF-beta and cytokines, that are important in the development of tolerance. So the milk is considered as a tolerogenic, inducing, that is important in inducing tolerance against whatever. And there's even studies that show, for example, if the mother is eating a protein, ovalbumin, the study was done with ovalbumin. It's a researcher from France that did this. They find that this ingested protein is also present in the milk. And because the milk is tolerogenic, when the newborns are eating drinking this protein with a tolerogenic liquid, that helps tolerance against this. So I guess answers a little bit of what you're asking. In terms of cross species, I think it's less clear. I don't think it's well known. Okay.
We saw that regarding the measurement of immune response, we can have almost no response or
That's
a super
right.
high one. Now do we know how to control the proportion of it's going to work, it's not going to work? Can we predict the outcome, or is it random until today? It's a mystery.
So how do we control the strength of immune response? Experimentally and in manipulation, you can. But the difficulty is when the question that she is asking, I'm allergic. How do I kind of reprogram the immune system to become less allergic? Or how do you reprogram a person that is autoimmune to not have that? I think to your question, we know a little bit. For example, we know what adjuvants to use. We know that the mRNA vaccine with the lipid motif, it will induce an immune response. We know that alum will induce. We know that others will not induce, and so on. But how do we manipulate the response that were pre-existing to the manipulation? Then is more difficult to know. And that's part of why these treatments with oral tolerance, it's very difficult to, it's difficult clinical trial because it's dangerous, and it's also difficult to, mechanistically we don't know well. So what did Nelson do? Kind of part of your question. That's the 1970 papers. This is measuring, I don't know if you guys even know this, it's sort of a plaque-forming colonies. It's sort of an ELISpot. Do you know ELISpot? It's like measuring a cell production of antibody specific to something. So this, people don't do this anymore. But you have animals that fed control, saline, and then challenge. That's immunization with ova. Then you induce an immune response. That is production of antibodies, in systemic antibodies to this protein. Immunization, that's vaccination. If an animal fed ova, the same protein, prior to the immunization, almost no response, 97% suppression of this. And it's specific, obviously, right? If you feed saline and you kind of immunize against another protein of egg, hen, heg, I forgot which protein is it, but it's another protein, HGG, you have anti-HGG antibodies. But if you feed over another protein, animal protein, and you immunize against HGG, you don't have suppression. So what you saw early on controls the immunization you're going to have. But what is interesting is what Nelson in the 1970s wrote in the conclusion. It's concluded that digestive route is extremely efficient in induction of immune unresponsiveness, which is the suppression of the immune response, which is maintained by the development of active suppressor mechanisms. Why did he say that? Because maybe what you have is just an absence of response. Why he's saying active suppressor mechanism? Anyone knows or can guess why he's saying this? Why he's saying it's active? What will be experiment would show is an active? There are two possibilities, right? You just, when you expose to the protein orally, you kill the cells that react to ova. There's no cells in the repertoire. That's was proposed for central tolerance. You delete the cells that react to ova. When you come to immunization, you don't have the cells. Then you're protected. And there's another possibility. So what would be your guess? Why he's saying it's active? So the experiment he did to show that is that he took cells from this animal here, transferred to a naive animal. Naive meaning never saw protein. Then immunized and saw that in this animal that received the cells of a tolerant animal, then also tolerance was established. So it was transferable. You can't transfer absence of something, right? You can only transfer presence of something. And then this was really a precursor of the field of regulatory T cells. It's very interesting in immunology. In the 1980s, everybody thought that tolerance was maintained by deletion of whatever reacts to self. And today we know that we all have self-reactive T cells and self-reactive B cells. But the way this is controlled, it's mostly by the presence of these self-reactive cells that control the immune response instead of trigger the immune response, the regulatory T cells field. Okay, so I'm going to show some experiments we did that are a little bit deep into immunology, but it's just an example of a thing that we addressed. Go ahead.
In the previous example, how it works? We have the T-reg cells.
Yeah.
They do not
How did it work the transfer here, right?
How it works for like when you transfer the cells, what's the mechanism that doesn't allow to?
Yeah. So what's the mechanism of transferring cells? This was only discovered, much later. But how, if you take the cells from this animal, transfer to a naive animal and immunize, how this is maintained? So what we all discovered was that upon feeding of ovalbumin, you induce regulatory T cells. These T cells further expand when you immunize. They become sort of memory, long-lived cells. If you take T cells from this animal here that get fed ovalbumin, in fact, people didn't know about CD4 and CD8 in this time. It was called LTI1 and 2, but anyway. It was before the discovery of CD4 and CD8. Initially, they thought it was CD8, then they saw it was CD4. If you take CD4 T cells that are regulatory from this animal here and transfer to a naive animal, these T regulatory cells will suppress the development of the immune response in the same way that they suppressed this immune response here. So it's an active suppressor mechanism. Is it clear? No?
No, I mean, I know they suppress.
Yeah.
So
How do Tregs suppress?
yeah.
Well, that's another topic because they suppress in a variety of ways. One of the ways that regulatory T cells suppress is here. Regulatory T cells can control the expression of co-stimulatory molecules by the antigen-presenting cells. Because they are specific to an antigen, they can control the initiation of the immune response to this antigen. So if they suppress a dendritic cell expression of these co-stimulatory molecules, they prevent activation of other T cells specific to the same antigen. That's one of the ways. They also secrete cytokines like IL-10, TGF-beta, which is immune suppressor. And there are many ways. Did I convince you? Okay.
Like it doesn't look like very efficient.
So it's very interesting why it's not efficient. So it's another topic, interesting topic. Okay, so, and that's kind of what we are addressing now in the lab actually. So if you have a dendritic cell here, right, antigen presenting cell here, you have a T cell here. If this T cell is regulatory, this T cell will regulate the activation of the dendritic cell. Normally, in the organism, you have many more T cells than dendritic cell in the draining lymph node. Dendritic cells, you can put in tissue culture 1 dendritic cell per 10 T cells, and that's sufficient to activate these 10 T cells. So they are different flavors. There's only one color, but there are different flavors of T cells surrounded here. So when you have an antigen, this is antigen A, this is ovalbumin. This T cell is specific to ovalbumin, okay? And this T cell suppresses the co-stimulatory molecule of this dendritic cell. Then, if this dendritic cell is presenting antigen to the cell B, or to the cell C, or to the cell A, this dendritic cell is now suppressed. So the activation of antigens that are not of exactly the same peptide that this T cell is specific is also suppressed if this dendritic cell is present. So that's how you spread the control to many more T cells than you have originally. There's an interesting experiment that is actually the first experiment that demonstrated regulatory T cell existence, genetic. It's Juan Afai, who was also my advisor at NYU. So Juan did an interesting experiment, and if I'm getting too specific, you guys say, oh, move on. It's exactly the example I gave about the multiple sclerosis, right? So Juan generated an animal that all T cells is a transgenic animal. So the TCR, you make it transgenic, all T cells recognize MBP. MBP is a protein of the myelin sheath, so these animals will develop multiple sclerosis animal models called experimental autoimmune encephalitis, EAE. However, in the generation of this animal, there is a process that doesn't matter for you guys much, but it's called allelic exclusion. So some T cells escape the TCR specific to MBP and become polyclonal. They recognize something else. So what Juan saw was the following. If 100% of the cells, if the animals lack RAG, the escape is not efficient. So all T cells will be specific to MBP. If all T cells are specific, 100% of the animals will have disease. We have EAE. I'm going to put multiple sclerosis. Okay. However, in the animals that it's allowed to escape the TCL specificity, if it's 98%, 98% of the cells. And here is animals. My handwriting is very bad, so you can see. Instead of 100% of T cells, only 98%, so only 2% were not specific to MVP. How many animals do you think got EAE? Just a guess. 0%. No animal got. Your question is very good. Why do you think Tregs are so efficient? This process seems very specific. 2% of Tregs control 100% of autoimmunity. So this experiment not only demonstrated that Tregs can suppress a majority of self-reactive T cells, but also demonstrate that it's a very efficient process. So we all have about 5 to 15% of Tregs circulating, including central Tregs, like auto self-reactive Tregs, as well as Tregs anti-protein to the diet and microbiota. So it's very impressive, the ability of these cells to control a majority of cells that are not Tregs in terms of inflammation. So this is a classic experiment, I think 1992, published in Cell. It's actually in the laboratory of Susumu Tonegawa, who discovered VTJ recombination and is now a neuroscientist. Okay. We went very deep into tolerance. See? Yeah.
Do you think this, I think this is a very surprising result.
Yeah.
I don't know this. So, does this tell us anything about the evolution of the immune system in particular? Because I think one thing that always bothers me about the possibility of the immune system evolving is precisely this ability, right, of avoiding.
Yeah.
About here, the system seems extremely robust.
Yeah. So I would say so people that study evolution of the immune system are very interested. The gene, so Juan almost also found the gene. So the gene was only found in 2003, 2004. It's called FOXP3. FOXP3 is the transcription factor that mediates the development of these regulatory T cells. There is a condition in humans called scurvy. Scurvy is severe and combined universal, whatever, autoimmunity. So scurvy are people that are deficient in this gene, FOXP3. They have autoimmunity against every tissue. Normally survive very little, very few years they die. In animals it's the same. Animals that are deficient die very quickly. So the appearance of adaptive immunity that allows this type of process that we are describing here occurred very close together to the appearance of also regulatory mechanisms that can regulate the inflammation. So as early as lampreys, which is the first jawed vertebrates that have RAG genes and MHC and have adaptive immunity, you have analogs of these regulatory T cells present as well. Because the regulatory T cells are also important in the process I was discussing yesterday of tolerance and repair, right? So they need to exist to avoid that is a positive feedback loop in the inflammation. Okay, so yeah.
Question more similar to what Benjamin asked. As it seems, we have immune cells that can respond to many things, including our own body, and most of the time they are just being downregulated by other cells. And my question, maybe it's ignorance of mine, I don't know, because I don't know much about the immune system, but for me, it seems that some kinds of allergies are very common, like peanut allergy.
Yes.
And I don't know, I have never seen allergy or something like that. Some people that have allergy to, there are thousands of different fruits, some of them seem to be very allergenic.
That's right.
Does this have anything to do with this downregulation process? And
it's unclear. So someone was asking this question, similar question yesterday, right? And I said, I think maybe it was you, no, someone. I said that's the main question in the field. of allergy, this question. Why do we start developing suddenly allergies, and why it's only certain types of food that you have allergy against? And what I mentioned is that it's a hypothesis. It's not entirely proven, although there is some demonstration, but some people also disagree. So it's just one opinion. That is Ruslan Medzitov, kind of using actual experiments that Nelson Vaz did in the 80s as well. So what they propose is that certain types of foods have a certain adjuvant. So the question of adjuvant is relevant. So nuts and peanuts and Brazil nuts and so on have a lipid content that can be misinterpreted in a way by the immune system as noxious. Noxious, not in the sense of pathogen, but noxious is something that will cause you, make you sick because you can't absorb. It will cause disruption in the peristalsis process and so on, right? Venoms and so on. And these types of food, we don't know why suddenly they become allergic, but they are more likely to do that than other types of food. But still, we need to answer why not all of us are allergic to peanuts, right? And this is a question that is not, in fact, we are trying to answer this question, I mean as a field, but there is an interesting tool that we developed. Gabriel Victoria is another Brazilian that is actually next floor to mine, and he developed something called lipstick. Lipstick is very interesting. Lipstick, the name was given after, but basically he developed a way that a cell, upon interaction. So this is what you're asking is very essential. We understand what is modulating. You guys ask, what is modulating what the dendritic cell tell the T cells, right? So what Gabriel did is the following. He did a system that the dendritic, that whatever cell, but let's say the T cell has a bacterial sortase that mediates a transfer of substrate to a cell to the receptor when it's very close to each other. So he developed a way that the T cell can label permanently the cell that interacts with. So the way it works is that labeling cell has the sortase, the labeled cell has the acceptor, the receptor of the sortase, and only when it gets below 10 nanometers, so very close, sort of immunological synapse, very close to each other, that is a stable interaction. It can transfer the substrate, which is a peptide, from this cell to this cell, and is a covalent transfer. So it stays in the cell after. So we can see the dendritic cell that interacts with the T cell and characterize them. So we are trying to understand for example, in a peanut context or whatever, allergy, what is being told to the dendritic cell to trigger this immune response. And a lot of this relates to this sort of mechanisms of tolerance that we are discussing here. It's very interesting what we found. I'm going to go to that, and then we can just discuss instead of going to more slides. So what we found is the following, and I'm going to try to explain. It's a bit complicated. In steady state, without immunization, we characterize the cell that is presenting dietary antigen. It's a subset of the dendritic cell that migrates from the gut. It's called DC1. Doesn't matter. DC1. DC1 are the cells that in the steady state will capture dietary antigens, migrate to the gut-draining lymph node, and then induce regulatory T cells. Why? Well, these cells are located in a specific region of the gut. They have ability to capture soluble proteins, and they have signature of expressing enzymes for retinoic acid, TGF-beta. So they are tolerogenic dendritic cells. So we asked, what happens when you have an infection? Which is part of what I would present next here. How can the immune system distinguish that infection, but you're also eating proteins? So why, when you have an infection, you develop allergy against everything? So it's very interesting what we found using the lipstick. We found that even during an infection, that's another dendritic cell here, the cell that is expanding during infection is another subset of the dendritic cell. It's a DC2. So let's say this is DC1, and this is a DC2. Okay? She should be showing here. Sorry. Okay. So what we found is the following. We found that in the steady state, this cell, which is telerogenic, they have ability of induced Treg, is presenting. Under infection, you have a large expansion of DC2, which are communicating with the T cell and inducing inflammatory cells. Inflammatory. And that makes sense because they have an infection, you should mount an immune response anti-infection. But the surprising thing came next. So we asked, well, what is happening to the antigen presentation? Is the dendritic cell presenting antigens to trigger immune response to the pathogen also presenting dietary antigen? The only way to know that is by doing lipstick because you don't know which cell interact with. And we found that during the infection, these cells are gone. There are no DC1 anymore. A particular type of infection, the helminth, these cells disappeared. And these cells are like 90% of the cells. However, the cells presenting dietary antigen was another subset of DC2, another DC2, that it was not inflammatory. So it was very surprising, and it's very immunological heavy, this explanation here. But what we found is that there is a way to separate, and we think that's because of the position of the cells in the intestine, that even during infection, that you have an expansion of inflammatory cells, you preserve the presentation of dietary antigens to another subset that is not able to induce inflammation. So that's a mechanism that is different than the regulatory T cell of preventing inflammatory responses to diet. And that's this is very recent, we don't understand everything, but that's an example of ways you have to regulate the development of anti-diet inflammatory. So that's why oral tolerance is so difficult to break, very difficult to break. For example, there are very few oral vaccines, very few. Polio is the main one, the Zegotinha. Polio was one of the main ones, and in fact, the oral polio vaccine is not very much used anymore because it's attenuated virus. In some cases, would induce reactivation of polio, but very few. And as a field, we think that oral vaccines are so difficult to develop because the natural result of ingestion of stuff is tolerance. So it's very difficult to induce an immune response to using a vaccination, but possible. But that's what we are trying to answer in this complicated scheme here. More questions?
So basic question, do we have when we ingest proteins or peptides,
yeah,
they are broken and you use it as a food. There is a way that we can have vaccines made of proteins, of peptides. How can an oral vaccine works?
So how can the oral vaccine works? It's very difficult. That's why we don't have oral vaccines. But the way that should work, we think, is to take this type of interaction here that induces the inflammatory response against pathogens and doesn't utilize of the mechanism that induces less inflammatory cells, sort of tolerance mechanism. So you have to target specific types of dendritic cells. That's how, and then you ask, how do you target specific types of dendritic cells? Well, they have, homing receptors. They have ways that they can bind. There are receptors that are only expressed by this cell, but not this cell, that you can target to develop improved oral vaccines. But the answer is not known, right? I'm saying the path, it's possible that is via this type of modulation. Because if you target this cell, you're not going to develop oral vaccine. You may modulate this cell, but the cell is normally steroidogenic. This cell is also not inflammatory. So you have to target specific cells that are inducing a response to a pathogen. But it's very difficult to break the absence of response to food.
I have other question that's
I think it's very basic too. We have the classical T alpha beta cells, and we know that they bind to peptides in MHC. Can you talk a bit about the T cells that are gamma delta?
Yeah, so that gets very immunology. Maybe it's not going to be interesting to everyone, so I can talk to you later, but very briefly answering, it's not very well understood the process of antigen presentation to gamma delta T cells. Alpha beta T cells see peptide, whatever peptide, MHC, for example. If it's CD8, a self-peptide or intracellular peptide with your own MHC class 1. If it's a CD4, normally outside from whatever diet, from outside of the cell, is captured, presented via MHC class 2 to a CD4. Gamma delta T cells don't recognize classical MHC. So they don't see conventional peptide MHC combination. Instead, in humans, there's a bunch of glycoproteins and non-conventional, what they call non-classical MHC molecules that is presented, but it's very little known. In fact, in humans, they know a little bit more than in mice. So it's a field that it's really, even though we know about the cells for 50 years, we still don't know exactly what they recognize.
Okay, I thought my book was old, but people really don't know about it.
They know some stuff that it binds outside the CDR3, the TCR, that can activate gamma delta in humans, particularly MICA1, MICA-B, but you know it's not developed field.
Okay, and another question, I like to think about like the optimality of like the biological systems.
Yeah.
And like in the epithelium of the gut, we have spaces between the epithelium cells. Like, wouldn't it be better like to have these cells tightly packed so the pathogens could not enter the body?
So the question of the tight junctions he was asking, right, in the back, was exactly that. So the epithelial cells are attached to each other via tight, it's called tight junctions, so it's definitely tight junctions. So the proximity of this is to make a sort of a barrier. However, it's not bound, right? Because you need flexibility because the cells are growing. They cannot be bound. How do you shed the epithelium? So it makes sense the way, I mean, of course it makes sense because that's what we have. But it's very tight together, packed together, but it's not attached to each other because once they grow, they have to fall into the lumen and go to the, be shed to the lumen. Pathogens that are invasive and efficient invasive, like Salmonella, disrupt the tight junction and they can invade between epithelial cells. That's a mechanism of pathogen invasion. So you normally don't have that. So this is in the case of pathogenesis, and that creates a lot of problems, of course. More questions? And we are about to be done. Did you guys think about anything that you want to discuss in the last day? I think Carolina will come tomorrow. So again, more, I think she's going to talk about her work on coronavirus. I highly recommend it because she's pioneering a lot of the stuff that you guys probably heard in the TV. And then we can discuss more like this, what I'm doing today, or we can discuss other topics that are whatever you may be interested. I mean, I cannot cover every topic, but science, politics, or recruitment of PhD programs, or how to write a paper. I don't know. Whatever you think would be useful time of our conversation. Any ideas or not yet? And it's okay to say to continue. It's also fine. I'm just, yeah.
Yeah, continuing would be completely great, but I think there's also, I think some of us would be very interested to understand the coming into an academic path in the US, as you both have done as Brazilians, and what kind of challenges Brazilians face and things like that.
So academic journey, that's one topic.
Maybe, I don't know, just a particular interest about aging, like the process of getting old and how this immunological process how can be achieved, like better health or
So
something.
that I may talk to Carolina because that's a topic that I don't work, so I cannot create a class on that. But Carolina works on aging a little bit in patients, so I'm going to mention that to her. Maybe she can cover tomorrow. In fact, it's very good timing because she just had a paper that actually, that's why she's not here. She's submitting the rebuttal exactly on that. She found that CoronaVac homologous booster in people above 50, that's not aged, but that's above 50, doesn't work at all in terms of inducing antibodies. But if you do a heterologous booster with Pfizer vaccine or AstraZeneca, then you trigger. So we don't know exactly why, but I think she's going to cover some of that. I'm going to mention that.
Yes,
to her.
just I say it's particular interest.
Sure. Yeah. Other ideas. I'm discussing with them on the Friday if they want to discuss something else in addition to this kind of to the topic.
Like immunology is a very experimental science.
Yeah.
Maybe could you talk about if there are any ways for quantitative approach using mathematics, physics?
Yeah, we can chat. I mean, I think there is you mean within immunology? Within immunology? The interaction between immunology and quantitative science? That's what I mean? Yeah.
If there are approaches possible, people doing that kind of work.
I mean, my ability to discuss would depend on, I haven't prepared anything on that, but, we can chat, but it's something that is not going to be, my contribution is going to be limited because, I'm going to say bioinformatics and computational biology, but then what else I'm going to say? It's the people, I know that there are maybe 3 or 4 people on bioinformatics here, computational biology, right? And in the US is the most disputed, the most, desired expertise. And I think it's going to get much worse or much better for whoever is working. For example, I'm in a grant with Kenya Honda in Japan, and he got this government grant. And in this grant, he proposed, and I think he's right, that in fact here they have a quantum computer cluster, I think. I saw in the first floor. Quantum computer is the only way that we're going to be able to absorb the type of data analysis we need. We are generating way more than we can analyze already now. In fact, with the decrease in Brazilian funding, a lot of Brazilians that are studying bioinformatics can data mine already what is published, right? And a lot of good publications come out of that. But I'm not sure if, I'm the best person to discuss that. I have some opinions, but I don't know if I'm going to say anything that is beyond the obvious. But we can try. I have 2 Brazilians bioinformaticians in the lab, for example, only doing that. And still not sufficient to cover the whole lab. And, it's very interesting because the biggest challenge, and I think we have to go, but the biggest challenge I think computational biologists track people have in the US is that they are still, in most cases, taken as someone to analyze data, and that's secondary to the paper. They are co-first author, for example, in the middle, or they are not the first author. And I think this will change. For example, I have a bioinformatician that was the first author because, it's the main. And then if you want to be independent, how this is going to be handled, because people still think that the wet lab is sort of the main data, people that's analyzing is sort of helping, and I'm sure this will change, because in many cases the analysis is going to be much more relevant than the source data. But anyway, this type of discussion we can have, but I don't know if, I can be very helpful to you guys on that topic, but that's my thought. Huh? We can, but how do I decide? I think what we can do is when we come, if Carolina can make on Friday, if not, we can start discussing and then should we change topic and then like that, right? Is that good? Because then we can cover different topics. We cover academic path and then, okay, it's enough, let's move to data computation or whatever. Is that reasonable? Okay, Carolina has an issue with the visa. She had to come, but the visa is not ready yet, so she may not be able to come Friday because she may need to go to Rio to get the visa. But for sure I will come. So we will have either 2 of us or only me on Friday. So should be fine. Okay.